Okay, so please welcome Shantanu Singh, who leads the Carpenter Singh Lab at Broad Institute, focusing on developing computational methods for analyzing microscopy images. Among others, his lab developed the cell painting assay in 2013, who now is widely used to test tens of thousands of drugs and genetic modifications to identify potential treatments. Today, he will share how such tools form a practical blueprint for AI in biology and drug discovery. Please welcome Shantanu Singh.
Great. Thank you. Well, thank you to the organizers for inviting me, and I regret not being able to make it for the first half of today, but I was able to catch up over lunch on the goings-on and the broad trends that we discussed today. And so I'll be talking about my lab's research, and I was asked to comment a little bit on how fast AI automation may happen in science, and I thought that to do that, it helps to first see a complete example, somewhat complementing what Carrie-Anne told us about in her research. So I'll start by introducing my lab's research, which is focused on using microscopy images for understanding how drugs and diseases affect cells, and then what discoveries that enables, and the impact that it's had on drug discovery. And then once you understand that landscape, I'll probably take the last 10 minutes or so to talk about what I think you'd want to hear about from scientists, which is looking at the same examples, same applications through the lens of automation, and what steps can be automated, what aren't, and what determines the pace of that. So the first part will give you the context, and hopefully the second will give you some insights about at least an N of one perspective on the automation as I see it. So, but just as a background here, I think it's worth sort of reviewing some of the technological leaps that have happened over time, and I think the community here would be quite familiar with this plot, the Moore's law of computing, which had predicted, roughly put, that the cost of computing would, or the speed of computing would double every 18 months or so, or cost would double or halve every 18 months or so, something like that. And that prediction, if you can think about it that way, held up for decades. And that's on a log plot, as you might know, and so it's even more impressive than might first appear. And if that seems impressive, then genomic sequencing would blow your mind, because what cost something like $100 million at the beginning to sequence a single human genome now costs less than $1,000, right? So that's also now on a log scale, very, very impressive scaling. But now the story from the drug discovery world is far less rosy. We fondly call it Arum's law, the opposite of Moore's law, which is that now it takes the cost of getting a drug on the market has been steadily increasing, and it now is very hard to get a drug on the market without spending close to $1 billion. I realized over time, after using this plot, using these slides a few times, that this has become a bit of a meme. I think this has sort of become a very not a wake-up call, but a call to arms in the drug discovery community. But for us folks who are on the tech bio side and using AI for drug discovery, this is the excitement that we have here, because we feel that AI could help speed up the drug discovery process and perhaps help reduce costs and bend this curve. This is actually a precursor, an older version of the plot that Neil showed us a while ago from Jayatunga. So this is actually from the 2022 paper of Nature Methods of Nature Medicine, and it showed that the investment in AI companies doing drug discovery was steadily increasing. You might have seen the investment world is a little bit less excited about it right now, but I think that broadly there's a lot of reason to be excited about the role of AI in drug discovery here. A lot of the drugs that we see here, all of the clinical candidates that we see here, are in the preclinical stage. But for the insiders, there's a lot of excitement, and I think Neil's more recent plot showed us that there is actually much more evidence now that AI-based drug discovery is something that can actually perhaps help bend that curve. So I'm going to focus just on one aspect of it today, which is using images of cells for this purpose, for discovering drugs. It's one of the many ways in which you can discover drugs. I believe last year's talk had folks from the cheminformatics side, where they talked about how you can use chemical structure to alone as a source of information to predict, to drive the drug discovery process. I'll be talking about imaging specifically. It's just one of them, my lab's research, my lab's focus, but it is one in which we have seen some of the most dramatic improvements if you've been following the computer vision field over the last decade and a half. There's been a revolution, right? And a lot of the deep learning revolution really started there, one could argue. And so that revolution, those advancements in computer vision have made its way over time into bioimaging, into the analysis of, into the methods for analyzing biological images. And it's now possible for us to analyze, to identify cells in an image and extract measurements from them and then correct for technical artifacts far more effectively thanks to these advancements that have percolated into our field. And all these advancements, many of these advancements have made their way into bioimage analysis tools that are produced by the community, including, for example, Cell Profiler that's developed by our lab, which is one of the workhorses for bioimage analysis in the field. And then there are many, many other such tools. Okay, so all of this, so deep learning in general has been a game changer in our ability to just quantify measurements, just extract measurements from images like this. Right? These are a sampling of biological images that that scientists produce, and they're very complex in terms of their variety and in terms of their structure. You see tissue images and you see time-lapse images. These are movies. You see 3D images, and some of them are fluorescent. I'll talk about that in a bit, and others are not. But all of these types of images, can now be relatively easily analyzed using a combination of classical image analysis methods, so to say, that have been around for the last 30 years, as well as the new modern deep learning methods. And now broadly, I think the thumb rule has become that if you can see a phenotype in an image, by phenotype here I mean the appearance of a biological system, right? If you can see that in an image if a human can see it and an expert can see it, then you can definitely quantify it, because it's come to that stage where it's almost certainly you can. And the thing is, if you can quantify that, if you can quantify a phenotype you care about, then you can now go and search for that phenotype, that specific characteristic, in a very large collection of image data sets, typically these very large chemical screens, for example, that can be run in pharma companies. And that is a game changer in terms of our ability to go and look for complex patterns in images, all right? So all of this is having real-world impact. Biologists are now able to use tools like this to go and look for patterns in assays and in experiments that they care about. Funding limitations, and increasingly now, obviously limit how many of the chemicals and potential candidates that we discover. So this can be pushed through the drug pipeline. But there's some really nice examples. I just pull out one where groups have used our systems and this idea of extracting information from images to get drugs onto the market. Now, all the examples I showed you, all those images I showed you, those are all actually cases where there were sort of one or two measurements that the biologists already knew that they want to extract from an image. Okay, so they look at this image and they say, you know what, I don't know, the image analysis is going to be hard, but essentially I want to be able to measure, for example, how far away are these specific organelles from the nucleus, or something like that, right? And then you now need to go and develop methods for that, either a deep learning method or a classical method. But sometimes they're much more complex. Like, for example, if you have metastatic cells in cancer, they are sort of these really strange-looking cells, and it's very hard to pin down the one thing you're looking for in that. Or if you have migratory cells that are moving around, hard to actually specify the specific parameter you want to measure. But instead, what you can take is a very different approach. This is actually something that you might be very familiar with, which is just a standard sorting approach in machine learning, which is, for example, like in your Google album or whatever app you use, if you want to be able to identify faces, right? The standard way of doing it is to just go and tag faces that belong to the person that you care about, and then you can go and train a classifier, or whatever the system is, to go and look for that person's face, right? Exactly the same thing can be done using a bunch of bioimage analysis tools that have been around for over a decade. There's the one I'm highlighting here is one called Piximi by our sister lab. The Simini lab, that is just a web-based tool. You can just run it in your browser, and it just uses your laptop or your computer's processing to do all the analysis. So there's nothing being sent to the server. It's all happening not just on your computer, but in your browser. So tools like this have now made it possible for us to again, quantify some very complex phenotypes that I've just, thrown here on the screen. You don't really need to read it. But essentially, just believe me that these are all very hard to actually specify as to what you're looking for. But you can, like they say, you know it when you see it. And you just see those examples, and you throw them into these bins, and then you go and ask these tools to go and train a classifier to go and find it, right? And then, again, this approach of this machine learning-based, like creating a classifier approach of finding a phenotype has also been very powerful, a lot of impact here too. And this is just one example where a company called Recursion had used this idea very early on in their formation to go and to go and find potential drugs for a cure of a rare disease. So good. So sorry for just like running through this. I wish we could talk about individual cases, but I think it's nice to keep this at the high level so that we can sort of transition into what I consider to be the next frontier, right? So all these examples, and you just want to think about this a little bit, right? All these examples I showed you of these biological images where we have specific phenotypes we want to look at, or specific examples of cells we want to look at. In all those cases, we are going in, we as biologists, are going in with a predetermined thing that we're looking for, either something we can directly tell as to what we're looking for, or examples of it, right? But we can take a very different approach too with images. Images are a rich source of information, and you can take a more unbiased approach. By that I mean you can take more like trying to find a signature of the cell as a whole, right? So just the way you can take a face and try to get a fingerprint of whatever, a profile of that face, you can do the same thing for cells in a very unbiased way, collect all the information you can, and then use that information downstream to go and solve problems. This is the standard, those who are familiar with terminology in machine learning, this is just a featurization approach. But when it comes to using it in biology, it's really powerful. It sort of links to the way, if you've heard of single-cell RNA sequencing and single-cell methods, all of those are essentially the idea that you can measure broad measurements across a wide spectrum of readouts from a biological system. And you can use images in that way, all right, as opposed to looking for a specific thing. So I'll just sort of slow down here a bit, and I just want to make sure you've understood this part of it, right? This part of the talk, because it sort of clues in a lot into the rest of the research I'll talk about. Okay, so the idea of image-based profiling, right? The idea of using images as a way of profiling a cell is pretty straightforward, and I'll just give you an example of what a typical image-based profiling experiment looks like. Okay, so imagine you have a set of chemicals that you want to, a set of compounds, chemicals that you want to profile. You want to get some sort of characterization of it. The way you do it in an image-based profiling experiment is you take those chemicals, and then you take a sample of cells, and you treat each sample of cells with a separate chemical, and then you let those cells grow for a couple of days, usually it's about 2 to 4 days. You let it grow, and then you go and freeze those cells. And then you go and stain them with some fluorescent dyes, these dyes that mark different structures in the cell, and when you shoot lasers at them, they glow. So now you do that, right? So now they're frozen in time, you have these fluorescent dyes, and then you go and image these. You go and image these cells under a high throughput microscope. These microscopes are amazing. They can just capture millions of cells in a couple of hours, right? So you go and image these cells that have been treated with these chemicals and have been stained with those dyes. And then, using the same image analysis software I told you about, you can go and, for example, identify each cell in that image, extract measurements from them, extract a whole bunch of measurements from them that read out not one or two things, but a whole bunch of things that relate to the appearance of the cell. And then for each sample now, you have this collection, this matrix of readouts, these high-dimensional readouts that represent the morphological state, the phenotypic state of the cell when perturbed with that chemical, right? And what you can now just do is that you take that matrix of information, and you can just summarize it in some way. Take the most trivial way, which is actually very hard to beat, is just average it, right? Just take the average cell, compute the average cell, and that average cell is really just telling you how did the cell feel when it was treated with that chemical? How did it appear? How did its appearance change with respect to a baseline state when you treated it with that chemical, right? So that becomes the image-based profile of that cell, that image-based profile of that perturbation. So what you've done now is you've taken this chemical, which was just this chemical structure, you've treated some cells with it, and you've mapped that chemical now to this high-dimensional phenotypic space, high-dimensional visual space, which represents how the cells change in the presence of that chemical, all right? And it turns out that this trick almost allows you to now solve a lot of problems downstream in drug discovery, and I'll try to walk you through some of those, all right? So just one preview here, since we mentioned cell painting, about what cell painting is, right? So now when you are, so those who maybe have some biology background, who maybe biologists, might wonder, like, right, so you're going and imaging these cells, you're marking them with these dyes that you told me about. What are these dyes? Like, you're not looking for any specific signaling pathway, you're not looking for a protein, you're looking for like global structure. So what do you mark, right? And so our approach was to say that we are going to pack in as many different dyes here that sort of stick onto different structures as we can fit into a single imaging assay. And what we came up with was what we call cell painting. It's a set of 6 fluorescent dyes that mark 8 major organelles in a cell, and you can image it in 5 channels. So you can think of it like most pictures that you can take on your phone are 3-channel images, right? These are 5 or 6-channel images. So you can image them in 5 channels that capture all these morphologies. So that's what a typical cell painting image looks like. It's a cheap assay. It's very easy to run. You can run it using a regular lab microscope, and these are fairly, it's a non-fussy assay, they just sort of dyes as opposed to antibodies. That's detail. And that has now made it, as a result, made it a workhorse of image-based profiling. It's widely used in academia and industry for routinely profiling chemicals and genes, which we'll come to in a second. Just to get their morphological fingerprint. Okay, so that's what cell painting is. I just want to mention that you can adapt this idea. You can adapt this idea, like what I told you here was you're marking all these major organelles, but then as we know, there are many different cell types, and sometimes some specific cell types might need you to mark other things. Like, for example, if you have heart cells, you want to mark something called sarcomeres. If you have fat cells, you want to mark some other thing that relate to the lipids and things like that. You can adapt this. There's nothing sacrosanct. There's nothing deeply optimal about cell painting other than that it is cheap. You can swap it out for other biology you care about. Folks at the Broad and around the world have adapted this assay for marking other structures in a cell, and you can mark heart cells and blood cells and neurons and things like that using the same strategy. Okay, so that we know now. We now have an idea about what image-based profiling is and what cell painting is. It's basically a way of characterizing perturbations you care about using images of cells. How can we use this information to speed up the drug discovery process, okay? And I'll give you maybe like 3 or 4 examples. Just keeping track of my time here. It's 17 minutes. And we can, for example, use, we could ask, can we identify signatures of disease and then screen for drugs to reverse the disease, right? So this is a very evocative example because you can just directly think of it in terms of actually being able to get a drug right away for a potential rare disease, and I'll tell you that in a second. So the idea here, I'll highlight, the project I'll highlight here is our collaboration with Mikko Taipale's lab in the University of Toronto. Mikko's lab is very interested in studying the impact of coding variation on protein localization. By that, it just means the ways that genes of 2 individuals may be different, how much does that actually impact the way a protein sits inside a cell, right? So that's the kind of thing that Mikko's interested in. So for example, if you have an image of LZTR1, this is one protein that's there in our bodies, and if you look at it, if you just mark that protein alone in a cell, and you look at it, that's what it looks like, and that's how it localizes. But now if you consider a variant of that protein that causes Noonan syndrome, it's a rare disease, it localizes very differently. You can just see it directly in the image, right? It localizes very differently, and it like sits in a different part of a cell, and that immediately gives you don't need a computer to tell you this, but you can use a computer to quantify it, and then right away what you have is what we can easily consider a screenable phenotype, which is to say that you can go and try to look for drugs that might revert that phenotype back to a healthy state, and that could be a potential therapeutic, right? It's a very simple, elegant idea. Probably too cute, and because sometimes that may not be the cause, it could be the effect. But at least it sort of gives you a starting point to start thinking about potential therapeutics. Mikko's lab went and did this for about 3,500 disease-associated alleles, and he found that and Jessica Lacoste his student found screenable phenotypes for about 200 of them, which is a small fraction of the 3,500, but it's a huge number when you think about the fact that you might need to go and do drug screens for each of these. Together with a bunch of friends around the US and in Canada, we are also now scaling it up about 20x times to do about 40,000 or some large number more variants. And now we are faced with a new bottleneck, which is how do we do these not just 200 drug screens, but potentially 4,000 drug screens, because you might want to do it for each one of these. And for that, what we turned to was this technology developed by the Neil and Blaney labs here down the road at the Broad Institute for screening them. And the idea is very simple for me to explain. It's a complex technology, but the idea is very simple. All you need to do really is that you take these 24 well plates. 24 well plates are essentially these multi-well plates. Essentially, you can just think of them as these large trays in which you have these small test tubes sitting there. Each test tube is called a well, and in each well you pour in cells from the same pool. And each pool is now being treated with one of the 400 or so alleles that we are studying, like 400 or so variants that we're studying, which is a mix of both the disease-causing variant, as might have been implicated, and the healthy form. And then you go and pour a different chemical. You can go and buy it from CVS or whatever, right? So basically, you just go and look for some drugs that are already approved, and pour it into each of these wells, and then you look to see whether the drug is able to revert the phenotype of any of the disease-associated alleles, any of those ones like the Noonan syndrome-associated allele, back to looking like a healthy state, right? And that's it. You can do this large experiment in a single large well. So now, of course, how do you now go and figure out which cell got which perturbation? I won't go into too much detail there, but essentially, this is very, very cool technology, and that's where Blaney and Neil Lab's work comes in, where they have a way of actually reading out as to which cell got which perturbation, which cell has which protein introduced in it, and also whether it was a wild-type form or the reference form, right? So that essentially allows you to do these very large experiments in a very compact format, and be able to read out these measurements rapidly. And we have a couple of experiments underway and a consortium that we started to work on this, and super excited. So we're hoping that in a couple of years we'll have something to show in terms of how this technology can truly scale for drug discovery. Another question we could ask is, can we identify gene function by grouping similar genes and alleles? So for this, gene function here is that genes do different things, and we don't actually know I think we probably know about a third or so of what, we probably know the answer for about a third or so of the genome, perhaps less. But for the rest of them, we really have either a fuzzy clue or no clue. But one way we could do it is like we did in this collaboration with Calico, was that we took the same technology that I just mentioned, that pool experiment, and we used CRISPR screening, essentially ways of systematically knocking out every gene in the human genome. And then we went and created these image-based profiles of those. That is, we were able to now map every perturbed cell where every cell has been perturbed by knocking out a gene, to some image phenotype, right? So now for every cell, we know, oh, this was the gene knocked out, gene X was knocked out, and this is what it looks like. And now if you go and do the same thing I told you before, which is aggregated and plotted in this high-dimensional visualization, we call it UMAP, you can now get a landscape, what you call a phenotypic landscape of what the impact of all the genes in the human genome were like, right? And that sort of leads to a deeper understanding, or not a deeper understanding, but at least it can help you start getting a deep understanding of what these unannotated genes are doing, all right? I'm going to try to skip over a couple of things here. I really should talk about this one because, maybe skip this one because it talks about mechanism of action. I'm going to skip over to talking about this idea, which is, can we go and try to find chemical regulators of genes and disease profiles? So typically, let's say you have a gene like YAP1, for example, genes that might be implicated in some disease. Typically, what you want to do is, when you have that, you want to go and try to find chemical regulators of that. You want to go and find something that modulates it, either amplifies it, turns it off. And the typical way it's done in pharma companies all around here in Kendall is that you go and try to design a specific assay for the function of YAP1, something that allows you to test how much of that YAP1 activity is happening. And then you go and screen your whole library of compounds that you have, 5 million, whatever you might have in your lab in your pharma company to go and see which one of those might actually change the effect of that, might perform in some ways that are modulating the effect of YAP1 in that assay, right? So it's a very complicated thing. The assays are cheap, that's great. Very often they're expensive. But what you can instead do is that you can take a very different approach. You can just take the same approach of image-based profiling. You can go and overexpress or knock out, that is, you can go and perturb YAP1 in a cell, in a sample of cells, and you can get this phenotypic, this visual signature of it. And then you can go and match that signature against a database of compounds that you've already collected. You can just think of this as like this very large face database or whatever, right? And you're just going to go and look for a match and say that, is this perturbation of YAP1, does it look very similar to something I've already done before in terms of a chemical perturbation? And if it is, then you can go and try to look for just the top matches, and you can go and test that in the lab. And you say, you know what, I'm not going to screen the whole 5 million compounds. I'm just going to screen this top 1% of matches that I found here, and then go and do wet lab testing. We actually did this ourselves, despite being a small academic lab, for about 7 genes, and we found promising modulators for three out of the 7 of them, something like 30 to 50%, depending on how you count. One of them turned out to be very promising for sarcoma, and that led my co-lead Anne to start a company based on that, just pursuing that one lead, right? So the idea here is, like, it's literally as simple. I mean, there's obviously a lot of detail in terms of how you do it, but the idea is really very simple. It's really trying to convert biology into this very large lookup table that allows you to generate hypotheses that might help you find drugs. I am going to skip over much of this. This is just to say that we have methods also that might allow us to predict some of these complex assays that I just mentioned, like that YAP1 assay, using images directly. That is, instead of going and doing the experiment of YAP1, we can try to predict the outcome of YAP1 assay using these images. If we have the time, I can get into it. But this was actually work that was done in collaboration with folks here at MIT who had actually spoken last year at one of your talks. I am just going to mention a couple of more things that may link us to the main topics of today, which is, all of this stuff that we want to do, all of these discoveries that we want to be able to make through this image-based profiling approach requires you to produce massive amounts of data. And we are a small lab. How do you now create a large reference data set for this kind of work? And our approach was to create a community in pharma, around this field. We launched the JUMP Cell Painting Consortium in 2019. This was a consortium of 10 pharma companies and several academic labs led by us at the Broad. And together we created the world's largest cell painting data set. It's a collection of 116,000 small molecules and 30,000 more that we threw in from before, and about 3/4 of the human genome that were perturbed. This is about 1.6 billion single cell profiles, and you can actually go and just download it if you like from AWS, thanks to their generous support of it, which would otherwise have cost us something like $40,000 a month to host and to serve. All this data is available now, not just for discovery, but for developing AI models to go and do these discoveries. Massive uptake in the industry and academia, right, a few months after this data set was released. We're also taking a very similar approach now to being able to do the same thing to predict liver toxicity, very narrow topic here, where we're trying to, Neil had shown that plot about toxicity and about failure rates, right? So a lot of drugs fail in phase 3. 30% of the failures in phase 1, phase 3, 30% of the failures in phase 1 of clinical trials is because of safety. And that basically just means that animal testing, which is a slow process, which has ethical concerns, but is also heavily inaccurate, needs to be revamped. And there's a lot of work there that's going on and trying to figure out new ways of doing it, new ways of solving it. We have created another consortium called Oasis, where we're taking the same approach, this very, very data-driven approach, to go and produce all these high-dimensional profiles using images, using transcriptional profiling, which I didn't get into, to see if we can use that as a basis for predicting toxicity in humans. So all of this automates. I just sort of ran through a whole bunch of examples as to what this can do for us in terms of 5 minutes, in terms of being able to help us with drug discovery. And there are many more examples. I'll put up these slides after the talk so you can download it if you like. But I just wanted to touch upon a couple of things, which is that, we're talking all of these things that we saw, gives you a sense of what cell painting enables and how it's already transforming drug discovery. But the real question isn't like just which parts can be automated, because as you've seen, much of this process already runs on AI. The question is, how can AI help us automate the AI-driven parts themselves. That is, how can we make these systems faster, more adaptive, and more autonomous? So I'll just share a few examples. This is vignettes from our own lab that we think are steps in the direction of automation that we think could make the biggest difference. I'll focus on just areas and tools that we have explored ourselves and just highlighting a few of them. So I'll be leaving out a whole bunch of them, so apologies to all our colleagues that I'm not highlighting here. One area is, I'll probably skip this one because this is sort of covered, but one area of automation that's quickly advancing is virtual staining. This technology has been around for a while, but it's been rapidly improving. So instead of like physically applying these fluorescent dyes that I told you about that goes and marks these organelles, you can now just infer these stains directly from bright field images, images that have not been stained with any fluorescent dyes, but you just sort of directly observe them under a microscope. And that may mean that we can eventually bypass staining altogether and letting AI generate the equivalent information automatically from raw images. So the work I'm highlighting here is from 2018. So just to say that this work has been developing for a while, and now it's come to a point where there are companies now who are routinely using this as a workhorse instead of using fluorescent dyes for imaging, right? I have to mention virtual cells because you must have heard the term virtual cells. I just saw this pop up in Time, which goes to show it's sort of getting into this somewhere in the hype cycle. It's sort of reached somewhere on, it's slowly cresting. In many experiments, we test how these perturbations change cell morphology, but these emerging virtual cell models, what they're allegedly doing is try to predict those outcomes directly. That is, just given the knowledge of the perturbation, that is just the chemical structure or the sequence of the gene, the models can simulate how a cell would look like or even predict the phenotypic response directly. And that's another step towards automation. That is, not only can you do those lookups, you can also potentially get those signatures. I will very briefly mention automating data analysis itself. I believe some of this was covered earlier today. We heavily have been leaning into this with some degree, with heavy dose of skepticism as well. Tools like Future House's Finch has been really insightful in how we can automate some of the data analysis we do. What we did here was just sort of throw in some data we had from a recently published paper and asked it to reanalyze some parts of it that we're curious about. It threw up some useful analyses that we were then able to follow up on later. And the other example is accelerating scientific reading, which is again here. This comes with a huge caveat that we just discussed right now of hallucinations, but we have found in general that there have been very effective ways of getting us off the ground in terms of doing this deep research for us up front, so that we kind of know roughly where we should look in terms of doing a literature survey before we delve too deep. I'm personally very bullish about AI agents as coders and builders for science. Claude Code is our workhorse right now in our lab. Claude Code is a really powerful agentic coding tool that we have found to be very useful. I'm going to put up these slides so maybe you can actually go and look at these repos yourself. I've personally used that myself now to just sort of come up with computer infrastructure in code, created these runbooks for our servers that are not just for humans. Actually, it's really more for AIs to use. Come up with some pretty simple theorems. We needed to prove something, and we were able to very effectively, not very effectively, but effectively come up with a simple theorem that allowed us to move forward. That was exciting because we thought that math might be a harder frontier to break with these LLMs, but it was somewhat successful. We're also building some simple tools from scratch that allow us to do very specific things, again, completely using Claude Code, and also rapid prototyping. We were able to, for example, very quickly convert some program from using CPUs to GPUs that resulted in like a 250x improvement. And the big gain there was just that we were able to do this in about 5 hours because we were able to just use the coding agent's ability to go and like poke around and figure out how to do the translation without getting too deep into it. So those are just some examples of how we've been using it. Just some reflections before I end as to where things I think are moving fastest and where I think we should be moving carefully when it comes to automation. On the left, these are areas, agentic coding and automatic data analysis. I think these are moving incredibly fast. The reason I think is pretty simple, as you might know. The people are building these tools are also its first users. And this tight feedback loop really means that progress compounds fast, and we in science can ride that wave rather than reinventing it. But translating all that momentum into science is not automatic. Science data is messy, data is noisy, and validation really matters. So even as those tools accelerate science, they also demand more expertise. And that you need to be able to still know when something is wrong, and that expertise is crucial when you're using some of these tools. Some areas like virtual cells, they still feel very early to us. The ideas are powerful, but in practice, simple models seem to be performing better. We're still figuring out what is genuine robust progress out there for tools like that. So overall, I'd say I'm optimistic, but very selectively so when it comes to the automation part. I do think automation wave is real, but the art will really be knowing where to trust it and how to use it to amplify rather than replace our judgment. So I think I'm over a minute. Thank everyone who was involved in this, and thank you for your time.
Hi, this is super exciting. Could you tell us about the robustness or the stability of using AI in this process, and what kind of practices can we use to improve the robustness?
The robustness of AI. So I think it definitely goes back to the same concerns that we discussed in the previous talk, which was someone needs to be doing the data curation part of it. All these 1.6 billion cells that I just mentioned that we have collected is beautiful data. It's a lot of data, but it needs very careful curation to figure out if there are systematic batch effects. So, for example, as you might have read in the news many times over, machine learning methods are very effective at capturing any pattern that there is in the data, including very spurious ones. For example, it turns out one very effective way of telling, of differentiating between two perturbations turns out to be as to which day it was actually imaged, right? So those batch effects turn out to be like the bane of our existence. So the robustness really comes down to some form of curation that is early on, where we annotate these data very deeply with all the experimental metadata that we have about it, and then use those experimental metadata to factor things out. But there are a bunch of other things, but that is certainly the one that is the biggest gain in terms of getting more robust data.
Thank you for the fascinating talk. Could you talk maybe a bit, you alluded to cross-modality or the mutual information between imaging and transcriptomic and other chromatin structure and things of that nature that are highly pertinent. How much can we actually learn from morphology, virtual staining? And the second question, if I may, can you maybe relate to the largest elephant in the room, which is discovery is just almost a negligible part of the entire capital expense of the whatever 2/3 of money going to clinical trial, less than 10% by some favorable measures of them actually end in a drug. So that Moore's law on the clinical side, what are your thoughts there?
Yes. So for the first part of it, the question was any insights on the multimodality, the complementarity of transcriptional profiling and imaging, and broadly here, the idea is just that if I can look at it, if I can look at a cell and take pictures of it, but then if I can also measure the amount of mRNA in it across all the different genes, is that information complementary or not? And the answer is somewhat boringly, there's some shared space and there's some complementarity in the sense that for a lot of, I didn't have the chance to go through this one example here, but essentially in this example right before, we'd shown that when you want to, for example, predict the mechanism of action of a drug, of a novel drug, and just use these transcriptional readouts as a way of predicting it, we found that for about 1/5 of the targets, we were able to find it uniquely only using transcriptional profiling, 1/5 only in imaging, and 1/5 were common in both. So it was like almost a cartoonish split. Like it seemed like this is exactly what one would mock up, but that's turned out to be the case. And I think broadly what we've found is just that in the end, it turns out to be a question of cost. How much can you spend on it? But I should address your second question, which is actually much more pertinent in terms of money spent, right? So that is exactly why we are focusing on the toxicity problem right now, right? I mean, the discovery part is great, and maybe getting better targets and phenotypic approaches would allow us to sort of speed up the pipeline. But the toxicity prediction part, we feel that if we can capture that, if we can predict a priori whether something is going to be toxic in phase 1, without without spending all that money getting into that, that would be to use the overused term, a game changer, because now phase 1 trials, you're able to not have these humans, first of all, take these drugs that are potentially toxic and instead be able to just predict it using these cell model systems. So we think that that could be, that could start actually directly getting into the clinical trials phase and allow us to cut costs there. Further down, I don't know. I think phase 3 and so on.
So
at that point,
efficacy
the point's up.
is unknown.
Yeah, at that point, I think you've already done the best you could early on, and I don't think these methods would help there.
Thank you.
Fascinating work. Christina McElhern, I'm at the University of Toronto working with the Acceleration Consortium,
Amazing.
where we're looking at automation and AI in materials discovery. And a pressing question for us is, what do the human complements look like? If we're building a lab to do this sort of work, are you changing whether they're more computer science trained or more automation trained? Are we teaching scientists to do this? Are the teams getting bigger? Are there more coordination costs? Like, what's the human side of putting this to work in discovery?
Yeah, no, that's a fascinating question, and it's actually particularly pertinent for me. Our own lab is a small but mighty group, as we like to call it. We have right now 3 postdocs, right? The 3 of us, 3 postdocs and 2 PIs working on this problem, and we have intentionally kept it small right now, just given funny things happening in the federal government. And so we decided that, okay, we're just going to stay light, and right now this has been a moment of reckoning for us in the sense that we have found that we are able to, just using some of these agentic coding tools, able to suddenly do a whole bunch of things that we thought we would have otherwise needed either more people to do or an intern to do or take a week off to do, right? So already when it comes to some simple tooling, like the ones I showed you there, that I think can be, I don't want to say replaced, but certainly heavily reduced in terms of the actual amount of effort needed in terms of human time. But when it comes to experimentation, I'm absolutely not the expert there. When it comes to lab automation and things like that, I hear claims every time I dread or peek into LinkedIn. I see huge claims about how lab automation is just a day away. I don't know the answer, but I know that labs like in silico medicine, companies like in silico medicine and others, Recursion included, claim that they're getting closer and closer to that loop, which is to say that you would still obviously need human experts to guide it, but then a lot of the automation would be a lot of the lab work could be automated. But I'm not the expert on that.
Thank you so much for this really interesting talk. I had a question because it seems to me that my understanding is that still a lot of this is at the predictive level, right? You're trying to predict the drugs. But now thinking about the science behind it, I was wondering if there is a sense in which potentially the integration of large language models with these visual modalities could be used to somehow at least express the grounds that these AI tools have for making a prediction, one prediction versus the other, so that maybe that could be a lead to us being able to find, describe in some sense the science behind these predictions.
Right. So getting to some sort of a mechanistic interpretation of it, and the field of ML and deep learning has a field called mech-interp, as they call it. The very same ideas, I wouldn't say the very same, but very similar ideas are being used in those virtual cell models that I just mentioned. And at this point, I've also stayed away from getting too deep into that literature, but from what I understand, the idea now is that they're combining LLMs. So essentially, they have these, if you're familiar with clip-like models, essentially language models as well as these visual models together to get a joint embedding, and then trying to then go back to the language space to infer what is actually being done there. So that's happening in the virtual cell models. But I am still waiting to see what the benchmarks say. I think benchmarks still say that you can't actually do too much better than average, just getting the average of all the perturbations. You may be able to explain it, but in terms of the actual performance, it doesn't seem to be that much better. But that is exactly one of the directions that the virtual cell folks and the biological, the tech bio community is going when it comes to virtual cell models.
I apologize for the really boring bureaucratic question, but
no, please go ahead.
Has the legal and intellectual property side evolved at all with this, where it seems like if we go to a world where drug discovery is incredibly cheap, then people can get the IP for it, with maybe no intention of actually developing the drug and then trying to sell it in a different.
Yeah, I do not have much insight into that, unfortunately. No, I mean, yeah, sorry, I don't think I have anything intelligent to say about it. It's an important question, though.
And I'm going to take the prerogative for the last question again. So your talk was interesting in the sense that you started with these computer vision techniques, right? And then you sort of finished with, coding and all of these other things that
Yeah.
could be beneficial. And I'm curious if you can just talk about, for your own, the kind of experiments you run,
Yep.
right? Like how much faster did your work get because of the computer vision stuff? And how much faster has it gotten because of the LLM stuff?
Yeah, so I would say that we're a bit of an outlier in the sense that our entire work is predicated on computer vision. I mean, the idea that you can identify cells in an image and extract features from them and featureize it is the basis of our science. So, we don't exist without computer vision. But so it is completely 100% dependent on that progress. I think to the extent that it's become faster, deep learning technologies have made it much more. Have made it much easier to solve some of the problems. So, for example, this problem of cell segmentation, which is, can you, given an image, can you identify the boundary of a cell or the boundary of a nucleus? It now is almost routinely the case that for all, I would say maybe 90% of the images out there, cellular images, you don't actually need an image analysis algorithm for it. You just throw it into something called CellPose. It's now become the de facto standard, and it just gives you the outline, right? So that is now, that is huge. Right? It used to take a lot of time to tune it. So there have been a lot of improvements in computer vision that have made things faster. But in terms of LLMs, the jury is still out. I mean, my lab, we have a sister lab. We have amongst us the complete spectrum of optimism to skepticism when it comes to this. And that ranges from reasons related to just that, sometimes people just haven't stuck with it enough and haven't tried it, to the fact that it sort of gives you, like our previous speaker said, a false sense of confidence that things are right. And it can just alter your own thinking. Like when you start, for example, that example I have up there of using Claude Code to change some code, once your coding agent has directed you in a certain direction, your own context window has now been polluted with the context that LLM is guiding you towards, and now it becomes very hard for you to steer back and think differently. So I would say that when LLMs, I'm very, very bullish about it personally, but I am very cognizant of the fact that some of my very, very smart colleagues who think deeply over the problem are very concerned about using these tools. So I don't think there's a consensus here.
Okay, interesting. Thank you very much.